good afternoon and welcome to the fourth session of this first edition of Technic MasterTalks. Today's session will be dedicated to the Department of Bioengineering. I'm Elise, a biomedical engineering master's student, so a student of this department, and I will be moderating today's session. We will have some time at the end for your questions, and after that, we encourage you to know more about each of the department's masters, as well as international opportunities, career development, and student support in the atrium right outside. I would like to thank Professor Patrícia Figueiredo for accepting our invite to be here today. Professor Patrícia holds a degree in physics engineering from Instituto Superior Técnico and also a PhD in neuroimaging from the University of Oxford. Currently serving as an associate professor in the Department of Bioengineering. She also leads as a principal investigator at the Biomedical Engineering Lab within the Institute for Systems and Robotics. Her academic careers includes chairing courses in medical imaging, neuroimaging, and neuroengineering, as well as holding positions of influence such as Secretary of the Scientific Council at Technic and currently as Vice President of the Executive Board of the Department of Bioengineering. Her research focuses on non-invasive imaging of human brain function using MRI and EEG techniques, alongside their innovative multimodal integration. Her work has been distinguished with a prize for Women in Science by L'Oréal Portugal and a Best Paper Award by the Portuguese League Against Epilepsy. She has held the title of Honorary Senior Lecturer at the Institute of Neurology from University College of London, while actively contributing to the European Society for Magnetic Resonance in Medicine and Biology. Her involvement ranges from membership in its educational committee to chairing the Congress Planning Committee for the 2021 ESMRMB annual meeting, and currently as President-elect. We will have the pleasure to hear what Professor Patrícia Figueiredo has to say about how bioengineering underpins key technologies in health, advancements in medical diagnosis and treatment, revolutionizing the intersection of technology and healthcare for enhanced patient outcomes and quality of life, and how we, as students, can be a part of this. Thank you, Professor. The stage is yours.
Thank you very much, Elise, for the introduction. It's a great pleasure to be introduced so nicely, first of all. So today I would like to take you through my journey in bioengineering, or maybe more specifically, and as Elise actually was tending to say, in biomedical engineering. And I'll do this by taking you through my views, which are by looking inside the brain and eventually trying to repair it. And by telling you about my own work, I hope to be able to give you some examples of how biomedical engineering has contributed for us to understand the brain better and also to treat brain disorders. And of course, these are only examples, but I will try to give you examples that involve master's and PhD theses as well. So for me, it all started actually with a bachelor's in physics engineering. There was no biomedical engineering at the time, and I was fascinated by the brain, but also by science in general. So I decided to study physics and engineering, and amongst many other things, I did study the principles of MRI at the time. So basically, that, as probably some of you know, if we have nuclei with a magnetic property or a quantum property of spin, they have a magnetic momentum that interacts with magnetic fields that we can apply to them. And because of that, they will have some interesting behaviors, they precess, and we can excite them. And by doing this, we can then read out a signal coming from them. And this is actually what we use to make MRI. And in particular, we use the nuclei of the hydrogen in water molecules that we have a lot in the body. So in like less than 30 seconds, I told you about MRI. And this was, it's a fascinating technique for many things, but in particular, to look into the brain. And of course, after finishing my physics engineering degree, I was really interested in the brain. So I decided, and I didn't have a biomedical engineering background, I decided to take at the time a program, PhD program in biophysics and biomedical engineering, which allowed me to learn more about things like anatomy, physiology, biophysics, cellular and molecular biology, all those other really difficult things after physics. And so I learned about places in the brain like the anterior cingulate cortex and the hippocampus, etc., etc. And eventually I went on to do my PhD, and finally I really started to work with this technique that I had heard about during my bachelor's in physics, and that was actually somehow synchronized with my own journey, because it was first presented in this paper in Science that I was showing you earlier in 1991, so basically at the same time as entering university. And in this paper, they showed for the first time that it was possible to look at brain function, as we can see here in color, using MRI completely non-invasively. So I could look at your brain in action without having to open it up. And this is quite common and standard today, but it was not 30 years ago. So it's relatively recent and amazing to me still. And this is basically what I've been doing since then. And one of the motivations that I had to, at my PhD, and I still have, to look at brain function was that when we look at the brain, we see that it is, this brain function is tightly coupled to blood flow. So what you see here are blood vessels going into the brain. You see the arteries and then also the veins draining the brain in a very artistic picture of it. And we know that this is really especially important in the brain, more than any other part of the body. And the reason is that the brain is very greedy. It takes up around 20% of the total oxygen of the total body oxygen consumption. And probably you know that our brains don't really weigh so much. They are around 1.5 kilos, and that's much less than 20% of our total body weight. So this is really a lot of oxygen consumption. And surprisingly, the brain cannot actually store oxygen, and it runs out in less than 10 seconds. So that's why if we stop breathing, our brain cells start to die very quickly. So it requires a very copious and stable blood supply to keep it working, and this is around 50% of the total cardiac output going into the body. So I think I've convinced you that the blood flow to the brain, or perfusion, cerebral perfusion, is a really critical biomarker of brain health. If it's not good, our brains cannot function well. And this was the motivation for a lot of people to develop techniques to measure blood flow in the brain and to image blood flow in the brain. Unfortunately, until now, actually, the conventional tools that we have to assess brain perfusion are not ideal. First of all, they usually require the injection of a contrast agent, so they are not totally non-invasive. And also, they are not truly quantitative, so they give us only a qualitative idea of brain perfusion. So this was my first project looking at brain function. It was more specifically looking at this blood flow into the brain. And I did this by leveraging on the physics of MRI and developing this technique called arterial spin labeling MRI. How does that work? Very briefly, but just so you see how I link the physics with the rest. So here is a sagittal scout scan through the brain. And I'm sure you all know that the blood vessels or the arteries are going through the neck until up to the brain to supply it with the blood flow. So what we decided, or people decided that we could do, is to take the spins, so those spins that interact with the magnetic field and that are in the water, in the blood, in the arteries, and label them magnetically using MRI so that we know they are coming from the blood. And this is something you can do non-invasively without having to inject a contrast agent. And then we just do this labeling actually by inverting their spin somehow. We wait some time for them to flow into the brain, and then we collect our image of the brain. And we basically created an endogenous tracer of blood flow by magnetically labeling the water spins. And this is quite a clever thing to do because it allows us to obtain these images of perfusion completely non-invasively. And what you can see in this movie over several slices through the brain, like the green lines there, is actually the signal we measure as time goes by. So you see first these bright parts here are the arteries that are where the blood first arrives, and then it kind of spreads out through the brain. So we observe some kinetics, right? We can see the blood arriving and then eventually flowing out. And this is very well described by tracer kinetics theory. So we can apply some mathematical modeling and then fit these measurements that we make to a model to finally be able to quantify perfusion. So we can make these images truly quantitative, unlike the other techniques. And that means that perfusion is measured in milliliters of blood per 100 grams of tissue per minute. And this is very important. We have a quantitative measure. We can compare a patient versus, for example, a normative data. And this is much better than what you can do with only a qualitative image. Okay? The problem is that it's not so easy as it looks like from this curve to estimate the models, the complex mathematical models that are underlying these. And our measurements, they are quite amazing, they are totally non-invasive and all that, but they're also very noisy. So nothing is as simple as it looks. And so at this point, I realized that knowing about physics was not, and engineering was not enough, and I had to get into some knowledge of biosignals and systems. And this is something that you as biomedical engineers already have, but I didn't have at the time. So eventually, and now I'm going to make a big jump to my time at Técnico, I started using some of these principles together with my colleague here, João Sanches, and with several master students that we supervised, and this PhD student also, Inês Souza, in collaboration with Siemens at the time, to develop methods for more accurately and efficiently performing this ASL perfusion quantification. So what we did was to employ a Bayesian framework in this case. Probably don't know what it is, but you might learn. By using this, we could optimize the times at which we were sampling the signal. So we could make the acquisition quicker, which is really important. Otherwise, the patient would have to stay in the scanner for too long. And also we optimize how we estimate these complex models using some sophisticated mathematics. And in the end, we could get these quantitative images with better accuracy and also more quickly. And we could also measure other parameters, for example, the arterial blood volume. So that's a second parameter that is also very important for us. And you can see here in color showing us where the bigger arteries are, so where we have more blood volume. So altogether, at this point, we were in the position to try and apply this non-invasive quantitative imaging technique in a clinical application. That's what we want to do in the end, right? So that was the work of my PhD student, Joana Pinto, and also several master's theses. And in this work, what we did, we had a project in collaboration with Hospital da Luz, where we studied patients with small vessel disease. This is a disease that you can have in the whole body, but when you have it in the brain, cerebral small vessel disease, it can cause lesions, as you can see in these structural images. And it's basically a neurodegenerative disease. It's the most common neurodegenerative disease. It's associated with age-related cognitive decline. It's also underlying most of the strokes and many types of dementia, including Alzheimer's disease. However, the structural lesions that characterize it usually appear only too late in the process of the neurodegeneration. And so the idea is that the blood flow changes in these blood vessels probably happen earlier, and they could be a more interesting, more sensitive, and earlier biomarker of these diseases. But of course, it has been very difficult to measure these perfusion changes robustly. So in this project, we applied our more efficient and accurate techniques to try and measure those changes in these patients. So what you see here are just the results of a paper where we show that compared to controls, patients had altered perfusion, but you see several bars here. So we couldn't just do it using a simple technique. We had to develop our technique further. And these developments included, for example, including a dispersion of the blood flow in the arteries in our model, which we didn't have before. But this is something we know happens more in the patient, so we had to account for that. Another thing is, this is a more complex model, so we start having more problems in estimating it if we have more parameters. So we had to also improve the denoising techniques that we applied, and this is the different color hues you see here. So this is just to exemplify how a biomedical engineer is able to improve imaging of blood flow, in this case in patients, by using more sophisticated modeling and signal processing techniques. More recently, we have also started to collaborate with Champalimau Foundation to apply this exact same technique, the cell perfusion imaging, in a preclinical setting. What does that mean? It means we are going to image not humans this time, but rather some animal disease models. And these are very important to be able to understand disease better, to study the mechanisms, and also to test, for example, new medicines. And in this case, we applied it to a stroke model. Actually, my PhD student, Sara Monteiro, who's been working on this, is the one who implemented the cell perfusion imaging in this scanner to image mice and rats. And she did an amazing job at getting very high spatial resolution images. The brains of rats and mice are really small, so you need very high spatial resolution, much better than in humans, to be able to see all the detail of the anatomy. And for example, she can see things like, of course, I don't expect you to know that, but I'm telling you that here you can see hippocampal layers or cortical layers and some local descending vessels that you could not see with the standard resolution. That's quite important. And then she applied this to an animal with stroke. And you can see here, hopefully it's very obvious, that there is a very abnormal perfusion, very decreased perfusion in this location. That's exactly where the stroke was induced in the animal. And again, with the higher resolution, we can define the area affected by stroke much better. So this is just, again, an example of how using engineering to improve our images, we are also able to study disease better. So perfusion was really, and is still really important for me and for us to study, to understand the brain. But actually most brain disorders are not just about a structural change, as I've told you already. Sometimes we don't see any structural lesions, or they appear only too late, or even a general perfusion change. They are more about how the brain functions, and there's something wrong with that function. And there are many examples of such diseases. You can think of psychiatric disease like schizophrenia, but also some neurological disease like epilepsy or migraine, in which you don't necessarily have anything wrong in the brain, let's say, in the structure of the brain. And so that brings me to the idea that we should be studying the function rather than just the structure. So as I said, maybe there's no structural brain anomalies, and even if they are present, they are not specific, so they could be associated with different pathologies. So imaging brain function could really deliver more sensitive and specific biomarkers of many brain disorders that we still cannot treat. Unfortunately, we still don't understand exactly how these brain function alterations occur, so we need a better understanding even before we can develop the markers. And so a lot of my work has also been dedicated to this better understanding of brain function and how it changes with disease. And again, using MRI. So again, the physics of MRI offers us a beautiful endogenous marker, in this case for the function of neurons, but it's actually an indirect marker, because what we look at is this so-called BOLD signal that stands for blood oxygen level dependent, fMRI. So how does that work? Well, we have a carrier of oxygen in the blood, probably all of you know, which is hemoglobin. And this carries it in an iron, I guess 4 irons for each molecule. And what we know is that when the oxygen is bound to the hemoglobin, the molecule is diamagnetic, which means it does not really perturb the magnetic field around it. But when the oxygen is released to the cells, so when it becomes deoxygenated, there's no oxygen there, the molecule becomes paramagnetic because of the iron's electrons. And so being paramagnetic means that it will disturb the magnetic field that we apply when we make an MRI image. And the spins that are in the presence of this magnetic field will feel that and will give us a different signal. So you see, the principle is that we can get a blood oxygen level dependent signal because the hemoglobin is giving us that, just like that. Of course, we have to tune the signal to be sensitive to this change, and we can do that. Why does this allow us to measure brain function? Well, because as I told you before, brain function is tightly coupled with blood flow, and this happens because there are mechanisms that we call neurovascular coupling that increase the amount of blood that comes to the tissues whenever the neurons are more active and need more supplies, namely oxygen and glucose. So in that circumstance, the blood flow increases. You see here a capillary, the blood volume also increases, and the oxygenation overall will also increase. So what we see when we look at this bolt signal over time, when we present a stimulus, is an increase in the signal. Okay, there are some more details, an overshoot and undershoot, but overall it increases. And this is exactly what we use to be able to do an image of brain function. We make an image of blood oxygenation, we watch over time when it increases, and based on that we know if the brain was active or not. To be more concrete, first we decide which function we want to study, for example, vision, and this is a visual stimulus that is very commonly shown to activate primary visual cortex. Then we design what we call a paradigm, in which you show the stimulus and alternate them with no stimulus, right, over time. Then you have to decide which part of the brain you want to image, so you prescribe a number of slices through the brain, usually the whole brain if you can. And finally, you pick a very fast imaging technique to be able to collect this 3D volume over time. So you see we end up with a 4D data set, lots of brain images over time, and in each pixel we have basically a time series, right, a time course of signal, which we then compare with the stimulus to check in which parts of the brain there was a signal change in relation with the stimulus. And this is the very basic principle of fMRI, which allows us to finally have the pretty image I showed you before, in which we see the activation of the visual cortex, here in red, overlaid on the brain anatomy. Okay, so this obviously is a very commonly used technique. It has been used to know study brain function a lot, but in particular I'll just show you an example of a very recent master's thesis from a colleague of yours. Maybe some of you know him, Alexandre Perdigão. He just finished, and he worked on a project we have precisely in migraine. And in this particular study, we are interested in understanding how the brain of migraine patients perceives pain. And we know that migraine patients, I think you all know what migraine is, right? These recurrent headache attacks that patients have, associated also with other symptoms. So these patients are known to have a hypersensitivity to different things and to pain. So we wanted to understand better the perception of pain in the brain of migraine patients. And we studied this interictally, which means between attacks. And that's because it's very difficult to study an actual attack, although we have studied that too, as I will show you. But the idea is that we really want to know how they perceive pain rather than how they feel pain, right? So the central mechanisms. And so what we did was an experiment where we asked the patients to imagine or to recollect their one migraine attack, one headache attack. And of course we had to alternate these with the control conditions, so we also asked them to imagine the relief of the pain. And then we also recruited a group of control subjects, healthy controls that didn't have migraine, and we asked them to imagine a very strong tooth pain. And this is because, I don't know if you know that, but it is the same trigeminal nerve system that is affected in migraine and also in tooth pain. So we tried to match as much as possible the two types of pain. But even if they were quite well matched, the perception of pain was very different between patients and healthy people. And in these three images, you see the regions that Alexandre found to be differentially activated in patients when compared with controls. And these regions, the inferior frontal gyrus, dorsolateral prefrontal cortex, and the anterior cingulate gyrus, are all part of what we know is the pain system. It has many different regions. And we found that patients actually deactivated these regions instead of activating them, which is what controls do. What was really interesting is that we also found that this deactivation varied with the frequency of the migraine attacks. So the more attacks the patient had, the more they deactivated these regions. So this was really suggestive that this mechanism must have to do with some maladaptation of the brains of migraine patients to pain. And of course, this is basic knowledge we are gaining about how these brains perceive pain, but it can also help us design better therapies in terms of modulating the brain activity of these patients and help them deal with this pain. one thing that is very obvious when I show you this, I hope, is that, okay, there are a few regions that I could name for you and that are identified, but we know that they do not operate in isolation. We know now very well that the brain works as a network, that there's a lot of connectivity between these different brain areas. And in fact, I told you all of these regions belong to a so-called pain system or pain network. So the idea that the brain works as a network, or actually as multiple networks interacting with each other, is something that is now very well known, but that came about after a lot of fMRI studies showed us that this is the case. And this brings us in another field of engineering, which is network science, that has been applied a lot in many fields, but also in brain science. So in fMRI, not only can we see that when you have a certain activation of the brain, it involves a network, but even if we don't have any stimulus or task, if we just observe the bold signal, right, so the brain function during rest, spontaneous brain activity. So this is just an example of how the brain signal is changing in different parts of the cortex. If we observe that these spontaneous fluctuations, we verify that they are correlated with each other with very specific patterns. So what you have here in this matrix is basically the functional connectivity computed, for example, using Pearson correlation, very simple, between pairs of regions. And you see that there's a structure. Some regions are much more highly connected with each other than others. So basically, we see a pattern that is not random of connectivity that people have started to call the functional connectome. And this pattern can be further analyzed to identify specific networks that are always there, even at rest, okay? So this is not something we observe when we perform a task, it's something we observe in the brain at rest. There's a visual network, for example, here, there's a dorsal attention network, visual attention network, somatomotor network, etc., etc. There are many ways of classifying them. This is just a quite canonical one that we have used. So it's very interesting that these networks are always there, but sometimes one is more active than the other. And these dynamics can tell us a lot about brain function and whether it's normal or not, for example. What you see here on the right is just a plot that we've come to love a lot, because it can be easy to see which connections are stronger and which are less strong in a simplified way. And the colors here indicate the different networks. And I'm showing already this because I will now give you one example where we use this network approach again to study migraine. And the idea is that we can study these networks even without having any stimulus or task being presented to the subject. So we could even study the attack itself, so when the patients are having the attack, without having any specific thing for them to do. So we can just watch their brain at rest and identify these networks. And that's what we did. We did this because we wanted to study the patients during the attacks. So we had to have some way of predicting the occurrence of the attacks to some extent. So we used menstrual migraine, which is very common. Patients often have crises associated with menstruation. And so we had to compare these with controls without migraine, but in the same phase of the menstrual cycle. So in our study, conducted by my PhD student, Inês Esteves, we actually managed to have patients scan during an attack, which is when they have all the pain, and also in these two phases, immediately before and immediately after the attack, pre and post-ictal, we call it. And we were careful to scan the controls in the same menstrual phase, as I told you, so the perimenstrual phase. And what we found by doing these types of analysis, just from rest, is that indeed something happens in the attack in terms of this connectome. So the functional connectivity increases compared not only to the other phases of the migraine cycle, but also to the same phase of the menstrual cycle in controls. So this is a very strong finding, because even when we control for the menstrual cycle, we see that there is something different. And this is just the result we have in this type of plot I showed you. And this you cannot see very well, but just to show where these connections are in the brain. and they involve specific networks, the visual, the dorsal attention, and the somatomotor. So this is very important for us also to be able to understand what's going on during the actual seizure. So yeah, fMRI is amazing, right? You see, I'm in love with it, and I still think, although it's been 30 years, there's a lot we can still learn from using this technique and keep improving it. It gives us excellent spatial localization, better than any type of image you can take of the brain function. You can do all these nice plots. However, remember, we're actually measuring blood oxygenation and using these as an indirect probe of the activity of neurons. So that's actually a limitation. And also, because this increase in the blood oxygenation takes a few seconds, we don't have a very high temporal resolution to detect the neuronal activity, which can be really fast. But we do have another technique that probably all of you know, which is EEG, electroencephalography, that gives us exactly that. It gives us excellent temporal resolution, and it's actually measuring the electric potentials generated by the action potentials and the synaptic potentials of neurons. So it's a much more direct measure of brain activity. However, of course, there is a problem that it cannot give us a good spatial localization. So we can get very rough maps of where in the brain this activity is going on. So when you look at this, you think, okay, we need to combine them, right? They are highly complementary. So that's exactly what we've done. We and some other people as well. So we have combined the EEG with the fMRI. It's definitely not something that is very easy to do, because there's a very strong electromagnetic incompatibility when you put this EEG cap with the electrodes and the conducting wires inside of the very strong magnetic field we use to do MRI. So it's very difficult to actually have good data when we collect them together. But we can do it. We can do it with engineering skills that I'm not going to go into. I just wanted to tell you that if we combine these two techniques, we can then use the temporal resolution of the EEG. So for example, we can and analyze something that we cannot analyze with fMRI, which are epileptic seizures and epileptic activity, which is a very fast activity of the neurons that is observed very well on the EEG. We can use the EEG to detect that, and then use the fMRI to localize where in the brain this epileptic activity is going on. And this is exactly what we've done in several cases. And I'm showing you here two examples that correspond to some master's thesis and then also PhD thesis from students of mine, Teresa Murte and Rodolfo Abreu. In the first case, we had a patient with a very specific kind of absent seizure. So this is a seizure where they actually don't move. They are completely absent without even realizing that time is going by. Although this is a terrible seizure, it's very convenient to study in the scanner because there's no movement. If you move a lot, the images are destroyed. So we could actually measure the seizure with both EEG and fMRI. So you see the seizure happening here. That's when the waves get higher. But we had no idea. You see, it's spread out in all the channels. These are the different lines, are all the channels on the scalp. You cannot really see where it is in the brain, right? It's everywhere. But when we do the fMRI localization, we can see that it affects specifically these brain areas. And in particular, the one you see here is the thalamus, which is right in the middle of the brain, and which normally we cannot really measure with EEG. So this is a very good example of how combining the two modalities gives us information that we could not have from either one of them. Another case that I quite like was this patient that we studied, a very young boy with a hypothalamic hamartoma. So he had a tumor in the hypothalamus, again, very deep inside the brain. And because of that, he had seizures, very, very bad seizures, many seizures a day. It was very serious. So we decided to scan him because it was not easy to remove this tumor. It was a very difficult thing to do for a surgeon because they cannot distinguish the tumor from the healthy brain tissues. They don't know exactly how to cut. So what they thought they could do was to interrupt the propagation of the activity from the tumor to the rest of the brain. But for that, they needed to know how does it propagate, in which direction, what pathway does it take. And of course, again, with EEG, we had no way of localizing this pathway. So we did, again, an EEG fMRI experiment, and we were able to see this network very well defined in space, and we could determine the direction of the pathway. So these are just 2 examples. Probably I should start finishing, but I'm almost there. So EEG fMRI gives us a lot of possibilities, and most of them, or most of the uses have been like this, right? We know some type of brain activity happening in EEG, but we don't know where in the brain it is, so we go to the fMRI. That's what I just showed you. But in fact, you could also think of the opposite, right? What if I know very well where the brain activity is, but I would like to be able to measure the temporal activity of this on the EEG? Can we do that? Well, for many reasons that I cannot go into detail right now, it's not as simple. And so for this, we need some machine learning. We need some clever machine learning to be able to estimate the EEG activity that gives rise to a certain pattern of fMRI activity. And this is the final example I'm going to give you. It's again the work of master's thesis in biomedical engineering, Marta Xavier and Inês Gonçalves, and there's more actually, people who have worked in this project. And in this case, why do we want to use the fMRI information to derive an EEG signal, right? So this is what we want to do. Well, it starts in the EEG in this case. We want to be able to use the EEG to do something we call neurofeedback, which is the idea that I have illustrated here, that we can train our brain activity if we get feedback from how well we are doing with our brain activity. So for example, if you imagine a movement, so if you perform a motor imagery task, in this case, the patient is asked to imagine moving their arms, and they are watching this scenario where they imagine they are actually rowing a boat. So the idea is that if we measure the brain activity associated with the movement, the somatomotor response, and we are able to classify whether the patient was moving or not, or whether they're moving the right arm or the left arm, and we provide this feedback to the patient or the subject, for example, in this scenario, by having the avatar arm move in the right direction, then we know that the person is able to regulate this activity and to learn to produce this response better. Why is this interesting? Well, there's a lot of reasons, but it's very important for neurorehabilitation, especially, and this is the case in our project, for stroke patients who have motor impairment and cannot benefit from conventional physiotherapy. In those cases, performing this kind of motor imagination or motor imagery tasks with feedback can help them promote neuroplasticity in the brain and actually recover their motor function. So this is the motivation, right? Why don't we just do it like that? And you can, but it doesn't really work for many people, too many. In many cases, this signal from the EEG is not sufficiently good. We cannot pick up the brain activity related with the motor task sufficiently well. If you cannot do that, you cannot provide the feedback, right? So it doesn't work. So we went to fMRI, and we went to fMRI. So we performed EEG and fMRI together, because if you perform fMRI during the exact same task, we can see very clearly the activity of the motor areas. So there's no question that the patient is activating the brain. It's just that we cannot see it in the EEG signal for some reason, and there are many reasons. But we thought, okay, let's collect the EEG, extract a number of features, for example, the power in different frequencies in different channels, and see if we can use a machine learning model to learn what features are associated with this brain network. And this is exactly what Inês did. She used, in this case, a linear regression approach using elastic net. And we have currently also some students trying some deep learning approaches to be able to predict better and better the fMRI activity of the motor network just using EEG. If we can do that, then we can really improve this neurorehabilitation technique. And I'm just looking at time. I think I'm just closing now. So I've shown you, I hope, that I really like these things, right? But the question is, why should you also like these things? Why should this journey be yours too? And so I tried to look up some numbers of why it is so important to study brain disorders. And this is just an example of one of these epidemiological studies that you can retrieve from the World Health Organization. But you can easily find that there's a very large proportion of us that will suffer from a brain disorder in our lives. Probably you all know someone who has some kind of disorder of the brain. And they have been shown to be the leading cause of disability in terms of years of life lost with disability, amongst all diseases, including cardiovascular cancer, etc. Right? So if you put all those together, so it's really important. And if you also estimate economic losses, if you are more into that, you can also see that they correspond to, or it's been estimated that the losses can be something like $5 trillion, and probably this is underestimated. So overall, this means that there is already, and there will be more and more investment in brain sciences, in the fundamental understanding of the brain, which is still lacking. We cannot treat brain disease if we don't understand what's going on in the brain, but also in developing technologies that help us deal with these brain disorders. And this is actually a very recent good news for us, which is that just recently in October this year, some European ministers signed this Leon Declaration on European Neurotechnology, which states a lot of principles on how we can and should develop neurotechnologies, so technologies to address the brain. And finally, we are here talking about bioengineering, and I think this is really the definition, or maybe more specifically biomedical, because it's about fighting disease. And it's already from 2016, this report from the MIT on convergence. But I quite like the ideas that I have in this report, and this is just a text from there. It's basically about the need to integrate biomedical knowledge with advanced engineering skills and expertise from physical, computational, and mathematical sciences. I think if you are in the bioengineering field, you probably all identify with this. But what I thought was really nice is that if you look in the report for what they think is needed for the brain in particular, then I found 4 points which I totally think we can do. One is new diagnostic approaches and non-invasive therapies. I already gave you examples of these. Another one is developing tools to quantify behaviors to be used in diagnosis, behavior modification. I'll show you that we can also do that. And the other is to explore connections between the brain and other organs, like the heart or the gut, very, very hot topic now. And finally, developing new nanotechnology carriers to get medicines and genetic therapies into the brain, which is obviously an area where our department is very strong. So I think at the Department of Bioengineering here at Technique, we can really address all of these. And I'll just give you some examples of colleagues that are professors in the department that are working on this. Professor Thanos Vorvopoulos, who in fact I collaborate exactly on this last project I told you about, works precisely on engineering non-invasive therapies. He works with these neurorehabilitation approaches using brain-computer interfaces and neurofeedback. These are just illustrations. I'm not going to tell you about these in detail. Professor João Sanches, sitting right there, is developing, amongst other things, tools to quantify behavior. So this was the second point. He, for example, has developed teleconsultation, augmented reality apps that allow psychiatrists to quantify behaviors based on these measurements of heart rate, head movements, eye blinking, etc. Then Professor Rita Nunes, also a colleague in the department, works with a lot of different advanced MRI techniques, in particular the heart, so she would be in the right place to explore connections between the brain and the heart, for example, also other organs. For example, she develops quantitative methods and very fast acquisition methods that allow us to have images of the heart that moves a lot, unlike the brain, in less than one breath hold, which is very important for us to be able to image the heart well. And finally, Professor Margarida Diogo, sitting there, who kindly explained to me a little bit about this slide, works on what I think is somehow developing new medicines and cell therapies. She works with engineering human-induced pluripotent stem cells, and in particular, they've done this really nice work on Rett syndrome, where they used this approach for modeling the disease. and showed that the physiological alterations that they found in these organoids that they were able to generate would allow them to screen for new drugs. So I really think this is a great department to be in as a professor and as a student. Of course, I have not really talked about everything that is done in the department. I've only really talked about my field, as I told you, and that's within the biomedical engineering area, with some connections with regenerative medicine, but not at all about biotechnology, microbiology, or bioengineering more generally. So please go and talk with the coordinators that are here to answer any questions if you have them, and I'm happy to answer also questions. Thank you.
So, Professor, I would like to thank you all. Thank you again for this very exciting talk. And I think I would start by asking the audience if you have any questions. Feel free to pose them in Portuguese if you feel more comfortable doing so. I think Professor Patrícia is okay with that too.
I think I still remember that.
So feel free to do so. Otherwise, I think I can start with one too. So, Professor, I think I would like to ask you how many of these technologies and techniques are already implemented in clinical practice, or else how long they usually take to be implemented?
Great question, and difficult one too. As you probably know well, in healthcare, indeed, technologies take a long time to be implemented, let's say, in the clinical routine, to have a prescription and associated with them. So I cannot give you a general timeline, but I can maybe say a little bit about the techniques I've worked with. For example, the perfusion imaging technique, ASL, which when I started working on it already a couple of decades ago, was really at the beginning, so it was completely research. And in this time, with all the developments that I showed you some examples of, and making it essentially faster, because if we present a medical doctor with an imaging, an MRI technique, and they love the image, but they say, how long did it take? And they say, 20 minutes. They say it's too long, right? They cannot fit it in the routine. So that's the end of it. So a lot of the time it took, it was to make it faster and faster while still keeping the information that we need from it, of course. And so now these sequences, these ASL techniques, are implemented in most new scanners, and so doctors can use them, and they are using them. I mean, the docs I collaborate with are definitely using them, although they haven't completely abandoned the contrast agent. So even when they do it, just in case, they also apply the contrast agent, except in children. And this is very interesting because, of course, the contrast agent has some adverse effects, and in children they really try to avoid it as much as possible. And they've started to trust this new technique enough that they say, okay, I will skip the contrast agent. So I think, yeah, maybe it took 20 years, but it's now a part of the routine.
Thank you so much, Professor. I don't know if anyone else wants to ask something. Okay.
Effects of the migraine pain in the brain. You showed a slide about that, and I would like to know if you could tell us.
Yeah, sure. I mean, this result is very recent, right? It's from a master's thesis, and we are still trying to find an explanation because it wasn't fully expected. And I think it's so what we know about the patients with migraine, as I said, is that they are hyper-reactive and hypersensitive to pain. So if you induce pain artificially with heat or some other drug, for example, they will have a much higher response than a healthy person. So this is known. So you would expect that their brain response would also be greater, right? That they would have greater activity when displayed with stimuli. And in fact, this is what happens if I induce pain in a patient with migraine. They have also a larger pain response. So what we did here was different, right? Because they were not feeling any pain. There was no sensation of pain. They were just recollecting the pain. So, and this is very well known about pain. You can probably imagine that we do have a somatosensory component to it, but there's also an affective, attentional, component to pain that is very important, that modulates our perception. And so what we try to isolate this part by doing only the imagery. And so we found that actually they didn't have a hyper response, they had a diminished response, so they were deactivating. So we believe that this is actually an adaptive mechanism that the patients develop to deal with the pain. So unlike the healthy controls, they are not activating this when they recollect the pain. So we don't know exactly what it might mean. We know, or it looks like it's related with the recurrence of the attacks because there's a correlation with that. And so we believe that if we target those brain areas and try to normalize, to have them activate them more like controls, we might be able to normalize also their perception of pain. But this is all I can tell you for now because it's really a very recent result that I think it's very exciting, and we are discussing with some pain experts to figure out what it means and how we can use it. Yeah.
So many people anymore say that pain is psychological. Is it true?
It is, and yes. Well, I guess I somehow, I don't know what it means to be psychological, right? It's not, it's in the brain for sure. I prefer to just talk whether it's in the brain or not, right? I don't know if when you say it's psychological, you mean it's not physical, but the brain is physical, right? And so, yes, pain perception is in the brain. There are many phenomena of pain, many chronic pain pathologies that I'm not an expert in, I should say already, that are very difficult to study because precisely we don't understand what goes on that causes this pain. Migraine is one such case, of course, because we don't know what starts the headache, right? So I think people are really struggling to understand this. We know that something happens in the brain when you experience this pain. Why some people are more prone to have that than others, we just don't know in general, but I mean, it depends on the specific type of pain, and yeah. But there's a lot of research, and this is, if I had the data there for pain-related disorders, that would be a huge chunk in that cheese that I showed you for sure. It's very, yeah, lots of economic loss as well.
You mentioned that one of the reasons you decided to study menstrual migraines was because you could predict when they were going to happen. How did you manage to do that with the little boy with epilepsy?
Oh, the boy with epilepsy is not menstrual migraine, okay?
No, I know.
Oh, okay.
Did you just
Okay. No, I understand. Okay. Sorry. Yeah, I understand your question. Very good question. Yes. So, yes, predicting an attack, either migraine or epilepsy, that is by definition spontaneous, is completely, well, it's very difficult, if not impossible. In the case of epilepsy, so in the case of the patient, so there were two cases I showed you. The first one, so these patients were selected by the neurologists we were working with. They were all children, and they were all having very severe, unfortunately, cases of epilepsy, which means they had a lot of seizures. So unfortunately for them, it was not so difficult to catch a seizure, but we didn't induce it. We just hoped in some way that it would occur during the period of time that they were in the scanner. Sometimes we didn't catch seizures, but in these two we did. So in the first case, it's interesting, we had the boy there for like half an hour, and the last minute he had a seizure, the absence seizure. So I remember this very well. It was already years ago. The other one, it was actually too easy because he was having seizures every minute or so. So we had actually, I think in the plot I showed you 5 different seizures. Having said that, you can still study epilepsy activity even if there's no seizure, because between seizures the brain already has some manifestations which are abnormal. We call them interictal spikes. So if you look at the EEG, you can detect some brief abnormal signals that are already related with the epilepsy. And so we can also study those. Even if there's no seizure, we can also study those. They are not exactly the full propagation of the seizure, but they give us already a very good hint that where it should be starting. So yeah, that's what we do also in those cases. But very good question, and sorry if I was, I didn't understand it initially.
In case of schizophrenia, how do you study that disease? Because there is no seizures or no pain, so how do you do it?
Okay, good question, for sure. So yes, well, first of all, even if there are no manifestations of the disease in terms of episodes, right, like what you just said, we can still question or investigate if the function, if a specific function is altered. For example, in schizophrenia, we know, and I have also worked in this, that the visual function is altered if you show a specific type of stimuli and you measure the brain response. It's not exactly the same as in a healthy volunteer. Also, auditory perception. So there are changes even in specific functions. However, this can be quite subtle and difficult to put in, let's say, in a whole brain picture. So I actually think schizophrenia is an excellent example for the importance of studying the brain networks. So in that case, we really don't think the disease is at all located in any particular part of the brain. They have alterations in visual function, in auditory function, in probably all functions. So it's not a localized pathology. And so what I try to exemplify also very fast, the idea that we can map these whole brain networks and analyze their dynamics and use this network science to extract different types of measures from them is very powerful for these types of disease where we don't know so much and we think it's about the whole brain. It's not just a specific part of the brain. Ok.
Desculpa. Ok. O que é que acontece com uma pessoa que não reage ao medo? Ou não reage a alguma coisa? Por exemplo, há um susto, há um medo. Essa pessoa não reage. Essa pessoa tem algum problema cerebral? Precisa de ajuda?
Não sei se sei, acho que não sei responder a essa pergunta. Também não sei se percebi exatamente o que é que querias perguntar.
Ou seja,
como é que se uma pessoa não reage, o que é que se quer dizer? Se não tem uma resposta no cérebro, é nesse sentido?
Mais ou menos a reação à felicidade, à tristeza, à depressão.
Emoções.
Sim, emoções. Essa pessoa não.
Ok, isso ainda é um outro domínio. As vossas perguntas só seriam bem respondidas se eu fosse uma especialista em montes, desde dor até emoções, mas é natural. Eu sou uma especialista em brain imaging, não é? Mas de facto é mesmo uma das coisas que eu mais gosto, é que nós acabamos por trabalhar em muitas áreas diferentes. Mas também sei um pouco sobre emoções. Acho que estamos a falar, se calhar, de patologias em que há alterações afetivas do afeto, não é? Portanto, aí entramos numa outra categoria que também são muito difíceis, também não percebemos, porque não há alterações estruturais, entramos mais uma vez um bocadinho no campo como da esquizofrenia, mas há muitos estudos, eu não conheço toda essa literatura, que mostram que de facto a resposta às emoções está alterada e isso pode nos dar pistas de como poder, se não tratar, mas pelo menos melhorar a forma de lidar com essas patologias. Sim.
Eu quero só aproveitar aqui enquanto também coordenador do mestrado, que tu desse um bocadinho a ideia de qual é o ambiente no teu grupo e no teu laboratório para quem esteja interessado em investigar estes temas. E eu sei qual é que é, mas era bom que tu
também
dissesses. E eu também estou lá.
O ambiente no meu laboratório é fantástico. O nosso laboratório é o Laboratório de Engenharia Biomédica do Instituto de Sistemas e Robótica, fica no 6º piso da Torre Norte, aqui no campus da Alameda do Técnico, e são todos muito bem-vindos se quiserem visitar, podem sempre escrever-me e podemos mostrar-vos mais. E somos basicamente, eu mostrei-vos, na verdade, os professores que fazem parte do laboratório, o professor João Sanches, que é o coordenador da mestrado em Engenharia Biomédica, a professora Rita Nunes, que é nossa colega na área da imagiologia médica, como eu vos mostrei, e o professor Atanásio Varvopoulos, que é o nosso primeiro estrangeiro no departamento e que é um especialista em interfaces, computer, o cérebro, computador, e portanto nós todos trabalhamos juntos. Eu acho que consegui, não sei se consegui, estou a dizer o que acho que tentei mostrar-vos aqui como as nossas áreas se interligam, cada um com uma especialidade um bocadinho diferente, e portanto o que nós fazemos é, temos um laboratório muito aberto em que os estudantes de mestrado e também de doutoramento, claro, têm um espaço. Nós temos uma sala de mestrados, de estudantes de mestrado, e portanto há muita interação entre estudantes de mestrado, de doutoramento, investigadores pós-doutorados e nós próprios. Temos um laboratório de EEG, onde fazemos as experiências de neurofeedback e de brain-computer interfaces, que vos mostrei só um bocadinho aqui. Não temos um scanner de ressonância magnética, não, infelizmente, mas nunca faria sentido termos um como os que há nos hospitais. Para isso temos várias colaborações, nomeadamente uma colaboração importante com o Hospital da Luz, mas não só. Mas vamos ter muito em breve um scanner para ensino. A professora Rita Nunes vai liderar essa parte, mas pronto, esse não vai servir para pôr lá o cérebro de ninguém, nem sequer de um ratinho dentro. Vai servir apenas para ensinar os princípios da ressonância magnética. Não sei se era isto que querias que eu falasse.
Que não disseste isso, que normalmente és muito generosa a colocar os alunos em interação internacional.
Sim, não falei desse aspecto.
Ou mesmo com
sim.
Outras instituições internacionais.
Posso acrescentar isso, sim, é verdade, que nós temos, todos nós, mas eu em particular, muitas colaborações internacionais, e temos tido, aliás, vários, se não todos destes alunos, especialmente a nível de doutoramento, tiveram parte do tempo no estrangeiro, em alguma colaboração, com, por exemplo, a EPFL, com a UCL em Londres, com o Politécnico de Milão, e em particular, e a nível de mestrado, temos recebido, de facto, muitos estudantes estrangeiros que vêm fazer as teses connosco. Eu não falei neles, mas tive 2 estudantes italianos, por acaso, mas um de Milão, outro de Turim, portanto, completamente rivais, não é? Que vieram fazer a tese neste projeto do EEG fMRI Modeling. E sim, temos tido, isto são exemplos, mas temos tido vários estudantes estrangeiros que vêm fazer ou teses de mestrado ou estágios. Neste momento temos no laboratório uma alemã, 2 espanhóis, se calhar estou a esquecer de outros, e os italianos, claro, há sempre muitos italianos. Mas também há muitos portugueses que vão para a Itália, né? Portanto, acho que se calhar há aí uma correspondência. Era isso? Sim.
Eu não sei se há muitos estudos sobre isso, mas relacionado nas doenças neurológicas relacionadas à dor, como é que o efeito placebo funciona no cérebro?
Há muitos estudos, isso eu posso responder. Okay. Não vou atrever-me a responder sobre algo que de facto não é de todo a minha área. Posso talvez dizer algo que está muito relacionado, que tem a ver, por exemplo, com a questão do neurofeedback. Não sei se vocês já sabiam, eu expliquei isto mesmo muito rapidamente, mas a ideia é que nós conseguimos melhorar a nossa atividade cerebral em algum domínio, por exemplo, a questão motora, com o neurofeedback. Muitas vezes coloca-se a questão, mas será que isto é efeito placebo? Portanto, tal como numa droga, qualquer modulação do cérebro pode ter um efeito por si mesmo ou por outros fatores. E, de facto, é muito difícil estabelecer isso, porque o controlo, não é como numa clinical trial, tem que ser afinado ali num ponto que é delicado, mas eu acho que é extremamente importante, e eu acho que é exatamente o que está a faltar nestas abordagens de neuromodulação mais tecnológicas e não tão farmacêuticas, se quisermos, em que isso obviamente está totalmente estabelecido, e em que se coloca a questão, será que a pessoa melhorou porque veio até ao laboratório, tinha já a motivação, estava ali concentrada e não sabemos. Eu posso dizer, aliás, isso é um estudo muito interessante que foi feito em relação à depressão major. Fizeram um ensaio clínico com estimulação elétrica profunda, como se faz em Parkinson, acho que toda a gente sabe isso. E havia uma hipótese muito forte de que, havia estudos de investigação que mostravam que os doentes melhoravam com a estimulação profunda de uma zona do cérebro na depressão. E decidiram fazer um ensaio clínico com controle. E não encontraram diferenças significativas. Portanto, havia um efeito, se quiseres, placebo, mas repara, os doentes de controle submeteram-se a uma cirurgia para serem, achavam eles talvez implantados. Portanto, eles melhoraram só por isso, digamos assim. e portanto acabaram por não conseguir que a técnica fosse validada para aplicação clínica. Mas é muito complicado quando estamos a falar de certas funções cerebrais que são muito complexas. Por isso, sim, é possível que haja efeito placebo, não quer dizer que não haja efeitos dos procedimentos, mas não sabemos onde é que está o efeito. No caso do neurofeedback, eu acho que é um dos interesses que nós temos, é perceber os mecanismos que levam a que haja uma melhoria e até que ponto são específicos daquele feedback que nós estamos a dar, ou poderiam até obter-se de outra forma. Ótima pergunta.
Okay, so thank you all. Thank you again, Professor Patrícia, and thank you all for being here. We hope you enjoyed this talk. And this is the end of our 4th MasterTalk session. Don't miss the next one, dedicated to the Department of Engineering and Management with Professor Ana Póvoa. You can already sign up in Eventbrite. And so we would like to encourage you, as Professor Patrícia said before, to go to the atrium where you can meet representatives from each master course of the department. Thank you and have a nice evening.